Direct from Milan, in Italy, you're listening to the ECTRIMS podcast, a scientific sounding board for the MS research community and the official podcast channel of the European Committee for Treatment and Research in Multiple Sclerosis. This October, approximately 8,600 MS researchers and experts from around the world have come together in Milan and online for the 9th joint ECTRIMS-ACTRIMS meeting. As always, the goal of this meeting is to allow the MS community to meet, share and discuss the latest research findings related to the pathogenesis, diagnosis and treatment of MS. Ideas will be raised, collaborations will be formed, and new research directions forged. Today we bring you the key highlights of Day 1 of MS Milan 2023.
Dear friends, dear colleagues, good morning and welcome to MS Milan 2023.
And much like the field of MS, Milan represents a beacon of creativity, innovation, and pursuit of progress.
This joint meeting marks a remarkable convergence of minds where the latest advancements in MS research and treatment are shared and celebrated. The continuous collaboration of ECTRIMS and ACTRIMS cannot be understated.
And without question, thanks to our entire community, MS is one of the, I think, 2 or 3 great success stories of modern molecular medicine. We're halfway home, but we're not all the way there. How can we potentially get there?
I'm Brett Drummond, host of this special episode of the ECTRIMS podcast. I'm on site here in Milan, in Italy, where ECTRIMS has wrapped up Day 1 of its 3-day Congress. Today I'm joined by Stephen Hauser, Professor of Neurology at the University of California, San Francisco, and this year's keynote ECTRIMS 2023 lecturer. Stephen, thank you for joining me.
Thank you, Brett. Delighted to be here.
So I just wanted to start with a little bit of a walk down memory lane like you did at the start of your fantastic presentation that you gave today. And you talked about being at one of the first MS conferences that existed back in 1982, where there were less than 50 people in attendance. And then fast forward to you giving the ECTRIMS lecture here today in Milan, where we have more than 8,500 people. That must be quite surreal. Having been involved in MS research for a long time, how does it feel being here at ECTRIMS today and seeing how things have grown over that time?
It's quite remarkable, and it's incredibly inspiring, not only for the interest that has grown exponentially in the work that we care about, but the progress that has been made that's helped millions of people worldwide. When I began my career as a young investigator, looking back, there were so few people who were interested in developing therapeutics for multiple sclerosis. There was even in many academic circles a therapeutic nihilism that this wasn't possible and that this would be bad for one's career. And to move from that place, which seems so archaic today, but wasn't that long ago, the MRI wasn't even a rumor in those days, to where we are now, with therapies that make a major life-changing difference for so many people. It's really quite remarkable. And the science that has been built also gives all of us optimism and hope that the battle will not be half won, but soon it will be fully won. We will have complete therapeutics for suppressing progression and perhaps prevention and cure.
I think you are living proof that it wasn't bad for your career to pursue this avenue. One of the quotes that stood out to me, and sort of following on from just what you said then, is that thanks to the entire community, MS is one of the 2 or 3 biggest success stories that we've got in medicine. But you follow that up also by saying we're halfway home, but we're not all of the way there. Sort of 2 parts to this question. Why do you think we have been so successful in MS and made all of that progress that you just talked about? But also, what challenges do you see as being the most difficult to overcome in the future?
Yes. I think that there were 3 major things that happened in our field. The first was the presence of physician scientists, an endangered species, but people who were walking the walk daily, connecting the laboratory to the clinic, and people who cared deeply about both science and the medicine. So that was one. The second was courageous leaders in industry and foundations who were willing to take high-risk gambles for therapies that may not have made commercial sense at the time. So those were two very big reasons for our success. And the third was a dollop of good luck, that as it turns out, the inflammatory part of multiple sclerosis arises in the periphery. And we could piggyback on advances in cancer medicine and immunology to deliver therapies to the peripheral body that didn't need to get into the brain in high quantities and make a major difference in disease. So it was passionate people, physician scientists, and low-hanging fruit, perhaps, compared with other diseases that don't have this type of peripheral inflammatory component.
The recipe for success in those three. And it's interesting you mentioned three things being important there, because the other thing that I think really stood out in your talk is when you were discussing MS as being a disease in 3 acts. And so for people who weren't at your presentation today and weren't able to catch it, I'd be interested in hearing you discuss that a little bit, but also how our understanding of those 3 acts has really changed over time.
Well, the 3 acts are the premonitory phase, what happens before people have their first symptoms. What is percolating? What's brewing? That's setting the stage for an attack on myelin and nervous tissue. The second is the early clinical stage of MS, when MS presents usually as attacks of disease caused by focal areas of inflammation coming from the bloodstream into the brain. And then the third act is that late progressive course of disease that supervenes in many patients after a period where they have relapsing, remitting disease. So those were the 3 acts historically. But what we've learned from the highly efficacious treatments that we have against relapsing MS is that progressive MS is not a separate entity, but it's there with us from the first moment that MS appears. We couldn't detect it earlier because MS often strikes young, healthy people with resilient brains. Patients knew that they weren't quite as good day to day, but we couldn't measure it. We couldn't record their disability that every patient knew he or she was having. So that's the historical part of MS. What we now know in 2023 is that the 2 clinical phases of disease, attacks and progression, are not separate bundles, but they both are present throughout the lifespan of the disease in most patients. So the first change was to bring together inflammation and neurodegeneration, relapses and progression in our thinking about the totality of MS. And the other is our exquisite opportunity using modern molecular medicine to understand the premonitory phase of the disease. We've learned from our studies that the earlier we can treat, the more effective our treatments are. So if we can get even earlier to the beginnings of MS, I think that that is one of the opportunities that we have to prevent the disease entirely.
It's really interesting in terms of how we've really built on, the foundations that you're talking about with those 3 acts have been there for a really long time in terms of understanding that, but how we continue to add layers on top of that. And obviously PIRA is part of what you're mentioning there, and we heard talks on that today earlier during the Charcot Symposium. And obviously we're going to get talks throughout the conference about what we're now learning about prodromic MS. It's really interesting and inspiring to see how we continue to build on that. And you must have seen that a lot over the years.
Absolutely. And as with most science, we want our data to be solid for the moment, but we also want better tools to overturn our conclusions. And that's what we're seeing.
It's very true. And I think the use of better tools, you presented some really fantastic data that I actually hadn't seen before you brought it up in your presentation, talking about a unique autoantibody signature that you've observed in a cluster of MS patients. Again, for people who may not have been able to be present. Can you talk us through that data and explain why it might be really significant for our understanding and potential diagnosis of MS in the future?
Absolutely, Brett. Happy to. This is work that was carried out by a large group led by Michael Wilson and Colin Zemechnik at UCSF. And it basically uses remarkable parallel display tools to let us interrogate or explore how the antibodies in our bloodstream and spinal fluid are reacting against every fragment of every protein in the human proteome. We have 23,000 genes. Each gene is making at least one protein, and those proteins are made up of amino acids. And in autoimmunity, the immune system attacks specific regions of specific proteins in our body. And this study used that kind of platform, really in a next generation way with the newest technology, and was able to use a data set that was collected by the Department of Defense and is still being collected, that's comprised of more than 10 million service people in the United States, beginning in 1981. It was initially started to look at the new epidemic of HIV, and they continue to collect serial blood samples from these service men and women along their lifespan. So there are more than 81 million blood samples in this repository. It's a great example of the importance of science that isn't always hypothesis-driven. Sometimes you need to just invest in something and build it without knowing for certain how it will be used down the road. That was how the genetics story of MS was opened up, because of these repositories. So Michael and Colin used this serum repository to select 250 people who were healthy when they entered the military, but years later developed MS, and wanted to ask if there was anything in their blood that predated the MS, but might have been a specific signal that MS was going to happen. And they found an amazing peptide sequence, an amino acid sequence belonging to about 54 of the proteins in our body. That is an unusual sequence and was responded to by antibodies only from the people who later developed MS, and by nobody who didn't develop MS. And then looking at the homologies or sequence similarities with other organisms, found that the closest connection was to an early activation protein of the Epstein-Barr virus. Now, currently, this can only be detected in about 1 in 7 people who will go on to develop MS. But improvements in the platform make me optimistic that we might be able to enlarge the proportion of patients who have a very specific MS signature.
Because this wasn't
years
found in
before
any other,
MS begins. It's
it
not
wasn't found
present.
in any other neurological diseases either, was it?
No, it wasn't.
Highly specific.
No, it wasn't. It's highly specific. And it gives us a way to perhaps identify people at very high risk for MS, as has been done in other autoimmune diseases like lupus, rheumatoid arthritis, diabetes mellitus. So this is very exciting. And it also adds to the evidence that there might be something very unique about the way that the immune system responds to Epstein-Barr virus that distinguishes people who go on to have MS from the vast majority of people who are infected with Epstein-Barr virus but don't develop MS.
This cohort of samples has been really valuable in terms of MS research. I mean, we learned a lot earlier this year with the Harvard paper looking at a similar cohort and being able to look at EBV exposure and the timing of onset of neurological damage. So it's really interesting what we're getting out of these cohorts of samples. And as you say, setting up the samples without necessarily knowing what you're going to use them for can be a really good thing to do. And I guess that also ties in with what you were saying before about earlier being better. Obviously, if we can start to identify people who are at high risk very early on, being able to get in and get them onto highly effective therapies at the earliest stages could only be beneficial to improving long-term outcomes.
Yes, it has become very clear that early initiation of highly effective therapy has better long-term outcomes, substantially better long-term outcomes than a traditional approach of starting with a lower efficacy treatment and then slowly increasing as needs arise.
Yeah. So one of the other things that I want to touch on from your lecture, because it stood out to me because it's certainly not something I think we would have heard 10 years ago, and it's something that I think researchers just by nature tend to be quite hesitant in terms of the things that they say. But you mentioned at the start that you think a cure is realistic, although you did say that we need to think about how we define that term. What did you mean by that?
I think that a cure for me means that all of the tissue damage that is happening from MS is halted. It's halted completely, permanently for the lifetime of that person, without the need for maintenance ongoing therapy. To me, that's the definition of a cure. Others have different definitions. A patient, very appropriately, might hope that a cure would also involve repair of earlier damage. But I think that a cure, to me, getting completely home rather than halfway home, would mean that this slow, insidious progression is completely arrested without the need for ongoing therapy.
Going back to where we started, thinking back to that first meeting in 1982, do you think back then that you thought a cure for MS would be a realistic outcome?
We knew so little in those days. The first treatment paper that I was part of described a population of people with progressive MS, whereas that population really had terrible relapsing MS, aggressive relapsing MS. Our understanding and harmonization of even the basic terms that we used was primitive. We were really at the beginning. It's worth remembering that in the late 1970s, if a patient came in with a problem that we thought might be multiple sclerosis, the neurology resident would ask the patient to get into a swimsuit. We would put the patient in a hot tub that was on each neurology floor and see if symptoms worsened. And if they worsened, we'd say, this is probably due to MS. We'd do a spinal tap. We would get abnormal immunoglobulins, antibodies, the products of B cells, and say, well, that makes the diagnosis more certain. But those antibodies are totally irrelevant. Those B cells are totally irrelevant. So we were at such a primitive stage, and it is magnificent to see the progress that has been made in a generation, and incredibly exciting to imagine the progress that we can make in the next few years with acceleration of scientific discovery and more support and awareness of the opportunity that this can be a curable problem.
I mean, it's fantastic and so exciting to hear. Just moving beyond your presentation slightly, obviously we've had a full day of presentations at the conference. I just wonder if you could touch on one or two other presentations that you've heard that really caught your attention today.
Yes, I think there have been many, and there will continue to be many, but the themes are so exciting. And the theme of crosstalk between immune cells in the brain, in the areas of smoldering tissue damage, I think is one of the great discovery stories of this past year. The concept that B lymphocytes and T lymphocytes are talking to brain macrophages, a cell called microglia, and activating those cells, which are then releasing damaging chemicals that are injuring neurons, injuring brain cells. So this crosstalk between the immune cells and microglia and the activation of damaging chemicals that are now clearly identified that injure myelin, injure brain cells, is a very exciting story because those are the culprits in the smoldering inflammation and neurodegeneration that are the key problem in MS today.
I couldn't agree more. It's been a really exciting day of talks. It's always so hard to choose which session you're going to go to with 9 different sessions running concurrently. As the last question before we let you go, you mentioned just before we started recording that somehow you've managed to find time to publish a book. Can you tell us a little bit about that? Because I'm sure people here will be interested in hearing about it.
Thank you, Brett. During the COVID lockdown, I had time for some writing. And the book is entitled The Face Laughs While the Brain Cries. It begins with a story of a patient of mine before we had treatments available who developed pseudobulbar palsy, which is a disorder where patients can laugh or cry without meaning to. And it describes what MS was like before we had therapies and my personal journey to try to find therapies for MS and how science and medicine have evolved over the past 40 years. So it's a memoir and also a story of discovery that I hope young people especially might find inspiring. And if just one person picks up the baton and decides they want to be a clinician scientist, it will have been well worth writing.
Yeah. Well, I don't think I consider myself as a young person anymore, but I'll certainly look forward to reading it. It sounds fascinating. Look, thank you, Stephen, so much for your time today recording this podcast with us.
Thank you, Brett. My pleasure.
And thank you to everyone for listening. Once again, I'm Brett Drummond, host of this podcast episode. I'm also co-founder of MS Translate, an independent resource centre that aims to simplify the complex wealth of information about MS for the greater MS community. The ECTRIMS podcast is produced and hosted in collaboration with 2 key partners, MS Translate and the MS Journal, a peer-reviewed international journal that focuses on MS, neuromyelitis optica, and other related autoimmune diseases of the central nervous system. The ECTRIMS podcast is supported by an educational grant from Novartis AG, Hoffmann La Roche AG, and Alexion Pharmaceuticals. Educational grant providers have no input on the podcast series content. You can read more about the topic of this latest podcast episode and more on the ECTRIMS website, ectrims.eu. Do subscribe to the ECTRIMS podcast on your favourite podcast channels and follow ECTRIMS on Twitter, LinkedIn, Facebook, and YouTube.